This animation shows brain development from conception through birth. In the first 4 weeks after conception, neural tissue known as the neural plate forms in the outermost layer of embryonic cells.
So this is a period referred to as neural induction.
The neural plate folds to form the neural groove, then curls to form the neural tube.
So in your book you see it looking something like this. It's a little better drawn, where if you imagine it in cross-section, that what you were just seeing, this outermost layer is referred to as the ectoderm, and then we have the mesoderm, which is here, and finally the endoderm. Now early in development, the mesoderm here sends out signals that cause the ectoderm to give rise or to form the skin, the epidermis, and the neural plate. So this area you see that's drawn in a red square is this area right here, which is the neural plate. And here you see mesodermal cells that have originated here, that have moved to midline and formed the notochord, which is the defining feature of the midline section, and which gives rise to differentiation of different areas. And laterally here, which again is up here, you see the somites, which give rise to skeletal muscle, tendons, endothelial cells, dermis, and cartilage. And what happens is this neural plate starts to roll up upon itself, closes, and then the closed portion branches off, migrates down to become the central canal. So before it closed off, it was referred to as the neural groove. And when it closes, it's referred to as the neural tube. And that then is what you see here. So the neural plate has folded to form a neural groove and then curls and closes to form the neural tube. And this again is a cross-section looking through that.
This tube differentiates into the forebrain, the midbrain, the hindbrain, and the spinal cord. The forebrain will develop into the cerebral cortex, which translates sensory stimulation. It also controls complex behaviors, thoughts, memories, and problem solving. The midbrain will develop into a neural relay station for sending information from the body to various sites in the brain. The hindbrain will control the most basic physiological processes, such as breathing and heart rate. The spinal cord is the pathway for conveying information between the brain and the rest of the body.
So the neural tube is formed by about the first 3 weeks of gestation, by about day 18. By 5 weeks of gestation, the 5 major areas are formed. And although they are not named here, you should know them. They're known as the telencephalon, that gives rise to the cerebral cortex and the basal ganglia, the diencephalon, which gives rise to the thalamus, the subthalamus, the hypothalamus, and the epithalamus, the mesencephalon, which gives rise to the midbrain, the metencephalon, which contains the pons and the cerebellum, and the myencephalon, which gives rise to the medulla oblongata. Know these: telencephalon, diencephalon, mesencephalon, metencephalon, and myencephalon. The way I remembered them when I was an undergraduate was tel, di, mes, met, my. Tel, di, mes, met, my. Now during this time, and up to about 5 months gestation, the brain is undergoing a significant amount of cell proliferation. In order to attain the cell numbers of 100 billion neurons, which are present at birth, Neurons are proliferating and dividing at the rate of about 250,000 new neurons per minute.
Between weeks 4 and 8, the embryo grows rapidly and the face becomes recognizably human. The eyes on the side of the head begin to migrate forward and two distinct hemispheres of the cerebral cortex emerge. Between weeks 8 and 26 of the fetal stage, the cerebral cortex grows to cover the midbrain. By week 28, a major structural change begins. The cortex expands greatly in surface area and becomes wrinkled and folded inside of the skull. Beginning at week 28 and continuing through week 40, the brain surface fills with hills and valleys, called gyri and sulci. These cerebral folds and wrinkles help to create a vast neural network, which allows the brain to become a highly flexible yet precise control center and processor of information.
So if you recall, when we talked about the inner membrane of the mitochondrion, the folding that occurs in the inner membrane increases surface area so that more ATP production can take place. And we alluded to that being the same principle as the generation of the gyri and sulci here in the brain. Imagine taking a piece of paper and crumpling it up. So you have a large surface area and you're able to crumple it up and put in a very small size.
By week 8, the structures of the brain are already in place, but the amount of brain tissue is rapidly increasing as many new nerve cells are formed. The formation of new cells is known as neurogenesis, or neural migration.
So we talked about the first phase, which was induction, and that's the specification of types of tissue in the central nervous system or in the nervous system. In other words, we have the neural plate, which gave rise to the neural groove, which then gave rise to the central canal and the neural tube, becoming more specialized to form the telencephalon, the diencephalon, the mesencephalon, the metencephalon, and the myencephalon. So here we can imagine the next step then being the proliferation of neural cells. And we talked about the rapid division, the 250,000 new neurons being born every minute, leading to about 100 billion neurons and about 1 trillion glial cells, non-neuronal cells. So here I might differentiate between neurogenesis, which is the birth of new neurons, and neural migration. What happens to the neurons after they're born? Where do they go and how do they get there?
During neurogenesis, new nerve cells migrate from the innermost layers of tissue outward. Like gymnasts climbing ropes, the young cells are guided to their final destination by fibers from another type of cell, the glial cell. Glial cells form the underlying structure of the nervous system. Although earlier formed cells rest closer to where they originated, later formed cells move further away from their starting point. In this way, the brain grows, adding layers to itself like the skins of an onion. Once each nerve cell reaches its destination, it begins to acquire characteristics specific to that area of the brain.
And it is at this point, when it begins to acquire characteristics specific to that area of the brain, we refer to that as differentiation.
At this point, each nerve cell sprouts the extensions it needs to communicate with other neurons. There are two types of extensions, dendrites and axons. Dendrites look like a network of antennas and receive incoming signals. An axon is a single, larger extension, which carries outgoing signals to other neurons. Synaptogenesis is the process in which synapses proliferate in the brain.
And so here we have the 5 steps that I'd kind of like you to think about. There are as many different steps to neural development as there are people who teach it. But in essence, this video has covered, and other people cover, 5 basic steps. The first being induction. The second is proliferation. The third is migration. The fourth, differentiation. And the fifth, synaptogenesis. So again, we have 1, induction, 2, proliferation, 3, migration, 4, differentiation, and 5, synaptogenesis. So remember those 5 different stages. And we'll go ahead and break each of those down just a little bit separately. So again, we have induction, the first phase, which occurs at about day 18, in which the neural plate develops, gives rise to the neural groove, which then becomes the neural tube. This process of the neural groove forming the neural tube is known as neurulation. The neural tube is generally closed between embryonic day 24 and 26, and the closure of the neural tube defines the end of the induction phase. During this phase, we have the establishment of polarity, what's caudal, what's rostral, what's dorsal, what's ventral, and we also have segmentation. Incomplete closure of the neural tube leads to defects such as anencephaly, a disorder in which the neural tube fails to close at the head end, which results in a major deficit in the skull and the brain. These infants die shortly after birth. Spina bifida, on the other hand, is a failure of closure of the neural tube at the caudal or tail end, and can result in herniation of both the spinal cord and the meninges up through that opening, just the meninges up through that opening, or simply a failure of closure. The proliferation stage begins at about embryonic day 24, which is when we expect to see closure of the neural tube, to about embryonic day 125, which is somewhere between 4 and 5 months. Pretty much your full complement of neurons will be in place. The next phase, proliferation, has to do with the rapid cell division of neuroprogenitors that gives rise to the eventual 100 billion neurons that comprise the central nervous system. So these progenitors, if you remember, we have the neural crest, which then closes over and becomes a neural tube. And if you remember, it initially was open, the neural plate closes over, and the ventricle then is just below, which was originally part of that neural plate. And so the proliferation of cells that become the eventual neurons occurs in the ventricular and subventricular area, which is pretty much right in here. This ventricle here gives rise to the ventricles in the adult brain. And the important thing to understand about this is that this area, this subventricular area, is also the area in which we see neurogenesis in the adult mammalian CNS. Now, this is an important issue that I want to kind of take a little bit of a segue on, because it was thought for many, many years until the early 90s that you were pretty much born with all the neurons that you're going to get. The neurogenesis was complete by the time of birth. Now, in 1992, Brent Reynolds and Sam Weiss published a paper in the journal Science, which describes the generation of neural progenitor cells from the adult mouse brain. This was absolutely a groundbreaking piece of work in the area of neuroscience. So again, until then, there had been a couple papers published suggesting that neurogenesis continues past birth, but nobody had really shown definitively that those cells actually reside in the adult mammalian CNS. So what these guys did was they cultured cells from the subventricular area of the mouse brain in the absence of the substrate, and that's important, in the absence of the substrate and in the presence of epidermal growth factor here. And what they ended up with were what they called neurospheres, which were these floating, proliferating cell balls that when they took and put on a substrate in the presence of other factors, these cells, which initially expressed nestin, which is a filament protein found in progenitor cells, these cells would differentiate into mature astrocytes and neurons. So the idea that you could actually generate new neurons in the central nervous system, in the adult central nervous system, was absolutely stunning. I remember, because we were very interested in doing this in the spinal cord lab at the University of Florida, and I remember meeting Brett Reynolds and Sam Weiss at the neuroscience meeting right after this article came out, and there were just maybe a couple, 2, 3 posters on generating neurons and astrocytes from the adult mammalian CNS. And the next neuroscience meeting, there were sessions upon sessions, there were talks upon talks on adult neurogenesis. It became one of the most important issues in the field and still continues to be that way. Now, since that time, there have been a fair number of labs that have shown neurogenesis in the adult human brain in postmortem tissues. We did so at the Buck Institute. We looked at neurogenesis in Alzheimer's brains. So I will post a review on those studies on the D2L site. Although people have shown that there is neurogenesis in the adult human brain, nobody's really definitively demonstrated to what extent, and it has been thought for a long time that it's only, a relatively small extent that you do, yes, see neurogenesis, you do see the birth of new neurons in the adult brain, but it doesn't happen a whole lot. And this has been pretty much held to be true until very recently. A very exciting study that just came out and published in the journal Cell. These researchers took advantage of the fact that there were nuclear tests carried out about 50 years ago. And what happens with nuclear tests, above-ground nuclear tests, is it increases the level of carbon-14 in the atmosphere, and then carbon-14 is radioactive, so you see an anticipated and measurable decay over time. And these researchers took advantage of that to look at postmortem tissue and show that neurogenesis occurs through carbon acquisition and deterioration of carbon-14. They showed that about 1/3 of neurons in the hippocampus actually renewed throughout the lifetime of the human. They, in fact, estimate that new neurons are born. There's about 1,400 new neurons per day, with that rate declining somewhat as the person ages. Now, these studies were just published, so it will be interesting to see, what the result is of these data. You have to kind of revisit maybe Alzheimer's disease and ask, is Alzheimer's disease really a failure of neurogenesis then in the brain, such that the neurons stop proliferating and you start having these buildup of plaques. So it's very interesting. It leads to interesting questions about learning, about memory, because the hippocampus is certainly the seat of those things, and paves the way for a whole new body of research. Now on to the next phase, how neurons get from the ventricular zone out to where they're going to be eventually. There's a number of different ways that they do that. One of the ways, for example, for peripheral neurons and for neurons that form nuclei in certain brain areas, these neurons migrate by virtue of cell adhesion molecules. So these are molecules that are laid down on the substrate that guide the neuron towards its eventual home. The other means of migration is an issue that we had discussed earlier, which is via the cells called radial glia. So these radial glia attach one of their processes to the ventricular layer here the cell body being somewhere along here, and send their other process out to the peel surface. And what this does is it provides a substrate for a neuron then, or a neural stem cell, to start migrating out towards the area where it's destined to reside. So here you have neuroblasts and radioglial that are being cultured together in a culture dish. So this is not in a brain, this is in a culture dish. And here you can see the neuroblasts migrating along the radioglial fibers. And this is a beautiful image from the cover of the Journal of Neuroscience showing these radioglia, here with their processes in the ventricular area going out to the peel surface and neurons migrating along these radioglia. And in the next phase you have differentiation, and that's the process by which a neuron, originally looking like this, starts to mature, continues to mature to become an adult neuron with its fate decided, whether it's a dopaminergic neuron, a pyramidal cell, a Purkinje cell in the cerebellum. That's what we call differentiation, is what that neuron matures into. And finally, we have synaptogenesis. Synaptogenesis is the making of connections between nerve cells. And while you see synaptogenesis throughout the lifetime of an animal, there are two periods of explosive synaptogenesis. That is in early development, which we're talking about here, and also in adolescence, where you see not just an explosion of synaptogenesis, but also what we call pruning, which is a dying back of those synapses that don't make connections. So there's a saying in neuroscience: neurons that fire together, wire together. And that means it's what we call Hebbian learning, which we'll get to when we talk about learning and memory. But it's essentially that neurons that make those connections, that fire and strengthen those connections, are going to be stronger and persevere longer than those neurons or those synapses that don't. And so eventually you have a dieback or pruning of synapses, and those synapses that die back are those that have not been strengthened by firing. So that brings us to the end of this video on neural development. It is far from the complete story. Please read your textbook, read that section on neural development, and we'll follow up more on some of this at a later point. Next week we'll move into neuroanatomy. The best you can say about neuroanatomy. It's very exciting, very interesting, but it's really a lot of memorization. It's really what is where and what's its function.